So our speaker for today is Professor Juan Perez-Mercader. So I would request Professor Rohit Srivastava to kindly introduce Professor Juan to the audience. Thank you so much.
Thank you, Neha, for this opportunity. So it's my pleasure to introduce Professor Juan. Juan Perez-Mercader earned his PhD in physics from the City College of New York. A professor de investigation in Spain's National Research Council in 1998, he became the founding director of Spain's Central de Astrobiología in association with the NASA Astrobiology Institute. In 2010, he joined Harvard as a senior research fellow in the Department of Earth and Planetary Sciences and Harvard Origin of Life Initiative. He is also an external professor at the Santa Fe Institute. He has worked in many areas of sciences, including theoretical high-energy physics, general relativity, non-equilibrium process, self-organization, complexity, astrobiology, and the development of in situ instrumentation for life detection outside the Earth. He was the architect of Spain's contribution to the NASA Mars program and its participation in the Curiosity rover. After joining Harvard, his work has focused on understanding the practical role of chemistry and non-equilibrium physics in the emergence of life to generate in the laboratory ex novo synthetic artificial non-biochemistry based forms of life. Thank you, Professor Mercader, for joining us. The stage is all yours.
Thank you. Thank you very much, Professor Srivastava, dear Rohit. And thank you, ACS, for the invitation to this chemistry series seminar. So now we are in business. Good morning, everybody. I am a newcomer to chemistry, but I have been working in chemistry in the lab with very talented postdocs and research associates like Rohit himself, and learned many things about chemistry. And what I've done is trying to understand how easy or difficult it is to generate a totally artificial, that is to say, systems which do not use any biochemistry, chemical systems that actually display all the properties of life. Why do we want to do such a thing? What are the main motivations for something like that? Well, there are many motivations for trying to make life in a test tube. Besides the motivations of the Frankensteins and all those, there are real technological and real scientific motivations to do that. What we are trying to do is somehow in resonance, in consonance with the actual development of a natural flight into artificial flight in the history of science and technology. People, human beings, looked at nature, got inspiration from nature when it understood how to do what they wanted to do, which is to fly, and then they started creating the technology that led first to flight gliding, and eventually to controlled powered flight by the Wright brothers at the beginnings of the 20th century, last century. That required understand first inspiration from nature, like for example, looking at pigeons. Required defining some mathematics, some mathematical models of how this flight could take place based on very fundamental principles like Bernoulli's principle, and then the application of such a principle by Lilienthal, and then of course the development of Lilienthal ideas by the Wright brothers, which eventually were able to implement a totally artificial flight. Today, a century and a bit later, the flight is incredibly developed in relationship to the first artificial flight machines. So this is one motivation, the development of technology, which is inspired in the technology in what we see in nature. That's actually a very important objective of scientific thinking and scientific work, which is unless looking at nature, seeing what nature does, and see if we can do it our way. So, but today, at the beginnings of the 21st century, or within the first quarter of the 21st century, ending very soon, next year, the quarter will end, we also have an extraordinary question that we want to answer. And the question that we want to answer is, does life exist elsewhere in the universe? And how do we think of that? Well, in the latter part of the 20th century, people started looking at the interstellar medium, the medium, which is between the stars in our galaxy, also in other galaxies, but in our galaxy in particular, and they began to discover a large variety of chemicals. They discovered that by using spectroscopic techniques and so on in the infrared, and today we do know about 400 different chemical species that exist in the interstellar medium. Among those species, for example, you have hydrogen cyanide, you have methanol, you have acrylonitrile, you have methyl formate, you have many other species. We also know, since 1995, we also know that there are many planets in the universe. In particular, in our galaxy today, we know that there are about 5,000 planets that we know of, that we have actually characterized. There must be many more. In our galaxy, there are 10 billion, American billion stars, and each of those stars, or many of those stars, could have 5, 6, 7, 8, 9, 10 planets like we in the solar system. Today, we are capable with new instrumentation, which is, for example, on board the NASA James Webb Space Telescope. We are able to look in detail, extraordinary detail, to what there is in the sky, and we begin to see in detail. This is actually an image, an optical image. We begin to see droplets, which are protoplanetary disks, which are planetary system information. So all this interstellar medium is full of chemicals, and all this, and there are many different types of planets and so on in our galaxy. So how did we then start here? We ask the question. We can ask the question: how in our planet, which is a rocky world, which is around here in this plot, and there are many more in other places, how in our planet did life start? We really do not know. We do not know the details, but we do know many things about the history of life in our planet. We do know that life in our planet was already present about 3.6 billion years ago. 3.6 billion years ago means American million, which is 3,600 millions years ago. And there was some living system, which we call last universal common ancestor, which is often abbreviated to LUCA, LUCA, which was already based on biochemistry. So biochemistry was already there. Before that, we hypothesized that there must have been some first DNA and protein-based life. We believe that about 3.8 billion years ago, there was on the Earth what we call the RNA world, a world in which the chemistry was dominated, the natural chemistry in the planet. Well, there was not artificial chemistry then. The chemistry in the planet was dominated by RNA in what is called the RNA world. Before that, we hypothesized that there must have been some pre-RNA world. We hypothesized that between 4 and 4.2 billion years, there must have been some prebiotic chemistry prior to biology. And then prior to that, about 4.2 billion years ago, we do know that there was a stable hydrosphere, that is to say, oceans in the Earth. The Earth formed about 4.567 billion years ago. During this time, between 4.2 and 4.0, we don't really know much because we don't have a fossil record that we can use to guide us. And we call this era here the protocell era. And there is lots of speculation. This era is potentially a place where there is a form of generalized biology, something that was postulated to exist or reason to exist by Bernal in the 1960s and others, including Oparin. We're talking about a biology which, without using biochemistry, implemented some functions of life. What we do know is that whatever happened here in this protocell era and whatever happened in the RNA world, etc., we do know that today, life in our planet, which is millions of species in the planet, is separated into three main domains: the archaea, the bacteria, and the eukaryota. This is the tree of life, and we can do phylogenetic studies and go and study where each of these objects, living objects that we see today, came from. And we infer from the and demonstrate mathematically and using computer models, and also computer models which accommodate very well the fossils that we see and that we have since this time about 3.5, 3.6 billion years old ago. We do know that all the living things we see here share a common ancestor. This is the time of Luca, about 3.6 billion years ago, or maybe a little earlier than that. So the question that arises is the following, what you see here. Did exist forebear lineages from before the most recent common ancestor? That is to say, are there common ancestors here? How difficult is it to make something that behaves like a potential common ancestor to this, Luca? And not only how difficult it is it possible that it was so simple that we could build it in the laboratory? So these are the type of questions that a physicist who is interested in chemistry is capable of asking, and in biology is capable of asking. It is important to remember that everything you have seen in the previous transparency, all the living systems that are represented very schematically in the previous previous transparency, all the living systems there use standard physical-chemical processes. There is nothing like a living spirit or anything involved in any life. What is involved is physics and chemistry. And of course, since the time of Luca, at least, the chemistry has been implemented via what we call biochemistry, the subset of carbon chemistry, which is the chemistry which is made of the chemicals which are produced by living and used by living systems. But between the chemistry, which probably was operating very early in the planet, and life, there is a huge leap in complexity. Chemistry is less complex in general than biochemistry. And then this jump from biochemistry to life is even more complex, or equally complex probably. But the jump from chemistry to biochemistry, how did that take place? This is not a question that I am going to answer today. This is a question which is still open, but we have put some little crumbs of bread in the way, so that those of you who are younger or who are older, but they want to get involved in this type of work, can actually do something about it. So we ask, and this is somebody who does chemistry, me, thinking like a physicist, which is also a part of me when I was younger. We ask the question, in physics, you actually tend to simplify, so to speak, reduce the natural phenomena into simpler phenomena. And what you often discover is that the complexity that you see in the natural phenomena that we witness, and where physics can tell things about them to us, there is always a very deep simplicity. That is to say, things are simple inside of them, that or they are controlled, they're regulated by very simple principles, which eventually, with the presence of time and evolution, in the case of life, for example, and the universe itself, lead to complexity. So Can we apply this widely held and extensively validated notion of fundamental physics that the complexity at the surface is a reflection of a very deep simplicity? Does it also hold for life? Well, let's explore that, which is what we will do in the next 20 minutes or so. So life today is characterized by many features, properties, like, for example, life is associated with order. Of course, everything that we see that is alive is a manifestation of biochemistry. And that is underlying any phenomena that we see here and we represent here. We see also evolutionary adaptation, like these seahorses in the coral. We see response to the environment. We see reproduction. We see growth and development. We see energy processing. We see regulation. And these are among some very fundamental features that are shared by living systems. It turns out that if you were to think like a very famous physicist in the 1940s, whose name was Erwin Schrödinger, who is one of the founders of quantum mechanics, he started thinking about what is life, what makes life tick. So he wrote a book which is called What is Life, which was read by many people. It's a fantastic book. It's a thin book. It's readable. And it's about physics, the physics of life and all that. But of course, without any of the answers that we know today. And some of the descriptions in the book are a little bit antiquated or very antiquated. But what we do know today, thanks to the work of thousands of people, is that all those properties that that there is a small set of properties that underlies every single living system, and that is all living systems handle information. That is an abstract quantity, and the information is handled with chemistry, is represented by chemistry also, and is necessary or is used in the process of generating itself, its transmission, its storage, and the use of information for control and memory, so that the system, living system's life doesn't need to reinvent the wheel each time somebody is born or some new system appears. All living systems, dear friends, perform something which is very important: metabolism. That is to say, they make their own parts, they manage energy that they capture and they harvest from the environment or generated by others, but they make they harness that energy and they make their parts. With their parts, they will end up constructing new parts for their self-reproduction. All living systems self-reproduce. Why do they do that? Well, a reason that you can think of is because living systems are open chemical systems. They are in communication with the environment, and there is dissipation. And with dissipation, there is degradation. And eventually, degradation leads to malfunctioning of the living system and therefore to death. So living systems self-reproduce as a way of avoiding the consequences of the arrow of time, avoiding the consequences of the increase in entropy as time goes forward. And finally, all living systems are capable of evolving. They evolve, they transform themselves, and they organize themselves in ways in which they can actually exist well in an environment in which they prosper and live. The evolution can be adaptive. The system reacts and adapts to circumstances, changes in the system, or this and this is very active on the part of the living system, or it simply is a failure of some kind, like for example, a mutation, and then the system readapts or in some way, or simply changes without needing to adapt. All living systems do something which is very important. They follow a so-called cell division cycle. The cell division cycle is something which is represented here in a very schematic way. This figure is taken from a book, a beautiful book from Marian Hunt about the cell division cycle, which was published in Oxford University Press in 1993. And in page 7, you see this graph. And the graph here represents in the left y-axis, it represents cell mass, roughly cell mass. In the in the right y-axis, it represents the amount of information, so to speak, the DNA per nucleus. In the x-axis, you represent time ticking, tick-tack, tick-tack, tick-tack. So the cell mass has to do somehow with metabolism. The DNA per nucleus has to do in some way with the amount of information that the system has. If you start your clock, boom, with the living system beginning, the first thing one thing the living system starts doing is increasing its mass, and it will increase its mass from a size of the mass 1 to 2 in arbitrary units, at which time it will divide. At the same time that it begins to increase its mass, the information in the nucleus, the amount of DNA in the nucleus, stays constant for a while. And then the DNA begins to be replicated. This is today, what we see today. And then it goes boom and jumps from the value 1 to the value 2, right here. And then it stays there, more or less constant. Many things happen there. And then there is what we know today, which is chromosome segregation. After the DNA replication has taken place in this period, there is chromosome segregation. The system stays with the chromosomes divided, and when the mass reaches value 2, boom, the system self-replicates. This is called the cell division cycle, and it does it over and over again, and it has children, and so on and so forth. So the cell division cycle is a way that nature in natural life, has found to integrate all these 4 properties, to unify all these 4 properties. And then you can ask the question as a physicist or as a scientist in general, chemist, can you unify all these functional properties into an aqueous, out of equilibrium artificial chemical system without using biochemistry? And that's the question that we will be addressing today. Part of the question is, and the approach will be to, part of the approach to actually answer this question is to first build a mathematical description of living systems, of these 4 properties of living systems. That mathematical description is something which has been produced in the literature, and it's like inspired in a similar idea, which comes from electromagnetism. In the 19th century, people, well, in the 18th century and 19th centuries, people began to see electromagnetic effects. And then people began to see the presence of Coulomb's law, the Coulombic attraction of charges. And they also began to understand that moving charges actually give rise to a magnetic field. And then there is the formulation of the Biot-Savart law. Mathematically, these equations describing Coulomb's law, the forces given by that, and in the case of the magnetic part of the theory, this is given by these other equations. It took the genius of James Clerk Maxwell in 1862 to actually put together a set of equations that are written down here, which many of you, probably all of you, know about, which are called Maxwell's equations, that actually unify the electrical and magnetic forces. They are called, the result is the electromagnetic theory of Maxwell. These equations, when you solve them in a computer, they produce solutions. They have solutions which are waves, electromagnetic waves, which were not known in 1862. And it took about 25 more years until 1887 for Hertz to discover an experiment that using an experiment inspired on these properties, an experiment that actually produced those electromagnetic waves. The electromagnetic waves 10 years later were used by Marconi to make the radio and so on and so forth. So here we are going to see if we can implement a unification, mathematical unification of this of the properties of living systems, just like there is a mathematical unification of Coulomb's and Biot-Savart's law in the Maxwell's equations. And we're going to try to see if we can do some experiment like Maxwell's, like the discovery that led to the establishment of the existence of electromagnetic waves by Hertz. And then we will try to see if we can think of what to do. So we did in applications. So in today's session, the first question we will ask is, can a generic mathematical physics and chemistry, high level, that is to say, very little detailed description of the properties shared by your living system be given? The answer would be yes, and I will show it to you in a minute. And then, could you use the solutions to those equations? It's not a computer simulation. This is solutions of these equations. Can you use those solutions to actually generate in the laboratory systems which are very simple and which do not need biochemistry to perform the 4 basic properties of a living system given here? And the answer to these questions is yes, to both of them. And that is where we are going to now. So in the next few slides, we're gonna just very briefly, very briefly mention to you the thinking that leads to these equations. And this will take 3 or so minutes, but it's important that I tell you the way a physicist or who is interested in these questions thinks. So the first point I would like to make is that we are thinking about the minimum, that is to say, the simplest possible that a scientist can think of, or a scientist like us, for example, can think of, which is necessary, the minimal set of mathematical conditions that will allow us to provide a mathematical, physical-chemical representation of the properties of life. So you need to take into consideration that you want to do this with chemistry eventually. So your mathematics will require a mechanism to transport molecules and another molecules for the molecules to react. So you will need a mathematical framework, which was actually discovered by Alan Turing in the 1950s, a mathematical framework, which is called the reaction diffusion equations, where there is molecules that diffuse and there is reactions that take place. We will need at least 2 substances plus a third one. Why? Because, but you can say, oh, but living systems use a lot of chemical substances, and the answer is yes, in the inside, in the details. But outside, you can characterize the living system as one species, one chemical species, which we will describe the inside of the cell. This is like the spherical cow metaphor. So the inside of the cell, which we will describe with the variable V. And another one, which is the food that the living system needs, which we will characterize with the name of the variable U. And then, of course, living systems produce waste. So that is important. And there is a minimum of 3 quantities, V, U, and C, 3 functions that we will need to input there. Living systems do not exist in the vacuum. Living systems exist in an environment, and the environment contains many things going on in it, and there is an intrinsic stochastic component, which we call noise. And so the living system will have to have, in its mathematical representation, will have to accommodate the presence of noise. With all these constraints that you have here in these 3 bullets, you can actually think up a model of a living system. And the model is something which is constrained by representing each of those functions that I told you, representing them by some reaction, diffusion, stochastic equations, and representing, imagine that you have a shell, a membrane of some kind, and then there is a net, some sort of lattice inside here, and in each of these lattice points, you do have a set of equations working. These equations are written down here. They were, if you are interested in looking at this, you can look at a paper by my collaborator Alberto Perez-Mercader and myself, which appeared in Physics of Life Reviews in 2022. It's an outgrowth of work done by Les Mes, Hochberg, Moran, and myself, which appeared in Physical Review Letters in 2003, and then it was enlarged in more work in 2004. Part of this work in 2022 consisted on the enlargement of this Les Mes et al. Work by adding the waste material, which is what will do something about the how the system is out of equilibrium. When you put these equations in a computer and you solve them in some net, here's what you get. The solutions that you get is you start with some very general boundary conditions, and then it produces those domains appear and they behave that way. You can actually look at one of those domains in more detail. And when you do that, you see this guy went from being spherical, more or less spherical, to going outside of equilibrium. Here, you have represented in this line here, in the blue line, you are representing the information in the system. And in the red and green traces, you are representing the mass. And you see how it changes as the system evolves, as the system changes with time. And you see that when it reaches a certain point, the system actually divides right here, just as natural living systems. So we can actually study solutions like this. It's a very complicated system, even though it looks very simple. Eventually, the system becomes very complicated, and you are only providing the very fundamentals. The important point is that all solutions that behave like this, or that behave like this, all solutions start by establishing a separation between the living system, the chemistry that will eventually make the living system work, and the environment in which it lives by a membrane. So this is a very fundamental initial condition. With that, you can actually, as I said, you can actually solve these equations, and you actually, the solutions to the equations follow in a analogous way what you actually see in the description of living systems through the cell division cycle. So can we actually build this? Well, we do. There are a number of obstacles that needs to be overtaken. One of them is that we want to translate the equations into a chemical network, which of course, at first we didn't know what the chemical network was or what the chemicals were, that can boot up. Boot up means that starts by itself, just like a computer. You boot it up. Well, a living system, when you are making it, or it should be capable of booting itself up without any external intervention. It should do it autonomously. And then it must sustain itself with that chemistry. And we want to think of life in a solvent, which is going to be water. Okay, why water? Because we see water and life associated in our planet. We could think of many other solvents, like, for example, liquid methane and alcohols and so on and so forth. So we want the system to actually do the cell division cycle, the implementation, the unified implementation by life of those four properties, to do it autonomously. And you want to start from a system, from something which doesn't have any order inside. A system that is completely homogeneous, and a system which is completely homogeneous is a system of high entropy. When the system does its chemistry, it will produce some order, and that will reduce the entropy. But in order for the system to be able to do, to carry out its chemical reactions, you need to solve two problems. One is called the concentration problem, which is that you are doing chemical synthesis, and the efficiency of any of these steps is very small. So when you combine all of them, you multiply quantities which are less than one, many of them, you end up with a very small amount of product, which may not be sufficient. So to solve that, that problem is called the arithmetic demon of chemistry, which was investigated by Sarah Tosa and Ireland in the 1960s and 1990s. You also need to be able to harness the information in the molecules in such a way that you can get some work out of the molecules. That's called the information problem. It turns out that these two problems can be overcome simultaneously by having a localized free energy gradient, which means that if you have a membrane, you have with something inside, you have, and which is open to the outside, not very open because otherwise it's not, it will not be able to concentrate enough. But if you have such a situation, then something very important happens. And that is that the system overcomes the presence of these two demons. But how do you do that in the computer simulations? Well, in the computer simulations, all the solutions that lead to this type of behaviors that I showed earlier, all the solutions that you look there have a beginning, which is with a membrane, as I mentioned already earlier in the talk. So how do we start? How do we do the membrane? Well, things looked very tough. And in the 1990s, late 1990s, developments in chemistry began to take place in the chemistry of polymers, began to take place. And one of the very important developments was the invention of raft polymerization. Raft stands for reversible addition, fragmentation, chain transfer, which is a form of leaving polymerization of the controlled radical polymerization type, which involves some free radical polymerization and so on, which is capable of with raft polymerization, you are capable of extending a chain in and the chains that you produce are of a very, very low polydispersity around 1, and which means that the chains are more or less of the similar size, as opposed to typical traditional radical polymerization where the chains are of multiple sizes. This is represented here by saying that raft polymerization gives you a very narrow distribution of molecular weights, and as opposed to regular conventional polymerization. This was, as I mentioned, invented in Chisiro in Australia, and there is a beautiful paper by Schiffer et al. In 1998, and there is also a patent by Le, Maud, Rizardo, and Thang, an international patent in 1998 where the process is described. This is extraordinary because using this and the property that all the chains are the same, one can try and make amphiphiles. Let's suppose that you start with a molecule, which like this one here, this part here, the blue part, which contains monomers of ethylene glycol. So this is polyethylene glycol, which is soluble in water. And suppose that you have a particular molecule here, which is called a chain transfer agent or raft agent also attached to it, which this contains sulfurs, and these sulfurs will actually be able to, if they encounter in the solution monomers, they will be able to transfer the monomers, attach the monomers, and extend this hydrophilic tail into a new block, which is hydrophobic if you choose the right monomers here. So what happens is that you can do this with photochemistry. And when you do it with photochemistry, this is what happens. You extend the chain, and then eventually the molecules that this part does not like water, the red parts like water, the blue parts like water. This is amphiphilic, this is hydrophobic. The molecule is amphiphilic, which means it contains both. Amphi in Latin means, and in Greek means both. it contains both the hydrophilic and the hydrophobic parts, and then they self-assemble. This was worked on using graph polymerization. It uses the fact that the chains are very much the same, and then there is a number of papers that were published by a group of different people, and some of the papers are given here. And then we make a contribution by dear collaborator and former postdoc Jan Szymanski and myself. We started writing this work, and you can see it in the transparencies when you get them and get a chance to look at them. To make a long story short, what happens is that by this process of chain extension using raft polymerization, the system self-assembles because it's unstable in the liquid, just like that. And then the molecules will minimize self-energy as free energy, and also they will consume energy in the system, and they can use food, like these monomers can be thought of as food also, and the system self-organizes into various structures. The various structures are very well known, and what happens, this is from the paper by a paper by Canning, Smith, and Arms, Macromolecules 2016, and it's described in absolute detail in a book by Israel Eshvili, who is the guy who thought about all of this before people began to do this actual implementations with chemistry. But in raft polymerization, you have the soluble stabilizer block, like for example, PEG, and then you have a monomer, which when polymerized will generate a hydrophobic block, and then through raft, it begins to grow and grow and grow. And then depending on the growth of this chain, you will have configurations of a space that look like this here, that lead, when these objects self-organize, self-assemble, they will lead to spheres, micelles. They will then keep growing. If you keep making this tail, hydrophobic tail, longer and longer, then this will lead to worms, which are actually cylindrical micelles, and eventually they can lead to vesicles. So this is what we were waiting for to see as an application or as a chemical realization of the solutions to the equations. We could use PEG, or we could use PISA, polymerization-induced self-assembly, because as the polymerization takes place, the system self-assembles in spheres, worms, and vesicles, for example, and it takes the system through a series of phase transitions, which are precisely consistent with the notion of the cell division cycle that I explained to you earlier, and that I showed to you in my transparency, in one of my transparencies earlier, where there was a growth in the mass and a growth in the a particular change in the information and duplication of information and so on in the system. So this we implement in the lab using a type of instrumentation which is very easy to have access to. We have there is a reactor here, which I will give a little bit more detail in a minute. A reactor here, and this reactor here is a cuvette. The cuvette contains certain materials. The materials are, in this case, are water, HPMA or acrylonitrile, for example, as a monomer, polyethylene glycol, a raft agent, and then we also use, in this particular example, we also use a catalyst to actually help the reaction go faster, which is ruthenium tris-BPY. And then we let the system do its chemistry. This pump here is to actually keep the temperature in the system constant. And then this is photochemistry, so you shine a blue light, which will activate the ruthenium and so on, and the reaction will start taking place. You can find this described in detail in a paper in Scientific Reports by Albertsen, Szymanski, and myself in 2017. We have developed this much more, especially in collaboration with Chen-Yu Lin and Sai Krishna Katla in my lab, and Kumar Siddharth and Keshwan Ren and others we have developed this quite a bit, and we now have reactors that we make using LEDs, strips of LEDs, and that we build, we put together in the lab, and then we actually do the chemical reactions, and we make sure that we don't use any chemicals which are biochemistry, in order to make sure that we are doing what we see is the consequence of the chemistry we're using. It's not a contamination of anything. And then what we do is we start the reaction, we take a sample, and the samples are shown, put in the microscope, and then we study their evolution. And when we make sure we don't have any biological material, we have this at the door of our lab. And what you see is the following. You see that this is the reactor. These are samples of the reaction in this picture here. This is time, this axis. So at first, the reacting liquid is transparent and is reddish because of the ruthenium. And then as time goes, with exposure to the light, the system eventually phase separates and produces what you will see in a picture, in a movie, in a minute. And what it produces is domains, very similar to the domains that we see in the computer solutions to our equations. And those domains do the following. This is not a computer simulation. This is not a solutions in any computer. This is actually a movie taken microscope. And we have hundreds of these movies. It's easy for us to make them. This is the bar here, is a 15 micrometer bar. This is the medium that, as it is deposited in the microscope slide, and after a certain time, when the phase separation took place, you begin to see this, and you see that there is a material which is produced, which is the generation through PISA of the vesicles with very important properties. And the important properties are seen here. You see that the vesicle is growing, for example, here. It's growing in size. You will see it now when I push the movie to continue. These are something like micelles. They're much smaller. They're growing. There is light, which is being shown here, and everybody's coming to the light. And there is also new systems that are being generated from the center. So let's look at this fellow here. And let me see if the computer simulation, the computer movie will work. So you see that this fellow, oops, excuse me. You see that that fellow is coming towards the center, okay? And it will do, I'm not gonna stop the simulation, the movie anymore, because otherwise it will flop. So they keep growing, but you will see what happens around here. And what happens around here is, as he goes, and many of them go, of course, look at him. He cavitates and he begins to grow again. And at the same time, others do the same thing. At the same time, there is an increase, a huge increase in the number of vesicles of the vesicles of the micrometer and larger size, so that you can see it with the microscope. We do know that there are smaller micelles and so on in the nanometer, in the tens and hundreds of nanometer scale. So we do see that there is an increase in the population. So what we see is the following type of phenomena. We see that they produce in the chemical reaction, they produce these cycles, they grow. This is one of them. It grows in diameter, collapses, grows in diameter, collapses, grows in diameter, collapses, grows in diameter, collapses, and so on and so forth. So we also see, when we count them, and we can count them with our microscopes, and this is reported, for example, in communications chemistry by Chen Yulin, Sai Krishna Katla, and myself, you do see that there is a change in the population size. You also see, as you saw it with your own eyes, that they move, and they move, and they form, they follow a form of chemotaxis. which is photochemotaxis, using the Marangoni effect, right? We don't have time to explain that. We understand very well how the system actually evolves and self-replicates. We understand how to do this with PISA combined with other chemistry, like, for example, with oscillatory chemistry. This is a paper which is the basis for another paper in which Professor Srivastava is collaborating with us, where we use the Belousov-Sabotinsky reaction to actually do the PISA. And with if you use the Belousov-Sabotinsky reaction, you see different behaviors, but still division and so on. You can, if you're interested in more details, you can look at the paper by Chang, Lin, and myself in small. It became the cover of that paper. And we have also shown, and I am like 5 minutes away from finishing, we have also shown that these systems can be made into populations. For example, populations where you have one member, one type of system in the population, like the ones here that look like a blanket flower here, which contain a catalyst which is sensitive to the light that you shine in the system. And you put food, represented here by the blue dots, and another protocells, which do not contain the zinc catalyst, which is sensitive to the green light, zinc TPP. So you see them when they have abundant food, they do this. And when they have abundant food, they both replicate. You see that there are more here of each of them. Of course, the one with the zinc replicate better, but if there is abundant food, nothing happens. But now, if you reduce the food, then something important happens. And that is that they begin to compete. And what we have shown, Katla, Lin, and myself, what we have shown is that if you take these two species and you count the number, the population of the species when they are grown separately, the ones with zinc TPP follow this path, this is time here, and the ones without zinc TPP follow this path. So when you put them together and they're grown together, then the ones with zinc TPP follow this type of path, and the ones without zinc TPP follow this path. This is precisely, exactly the same behavior that was discovered by Gauss in 1934, and to show that there is competition in life. And he showed that Paramecia, if you put two species of Paramecia that share a food, just in our example, then the growth curves for the one who wins, the one which is better suitable, which is called Paramecia aurelia, follows a curve which is similar to this curve, and the Paramecium caudatum follows a curve which is similar to a curve in some scale. And when they are grown together, they do this, which is precisely the annihilation of one by the other. You have a nice representation of this here, and we know that this happens because the ones with zinc, which are these ones here, grow much better. They all degrade, and the ones who stand degradation better are the ones with the zinc, and then they actually even take probably food from the parts of this one, which actually expels some partially reacted amphiphiles, and which can then be used as some seeds for molecules. We have also some seeds for new living systems, new artificial living systems. We have also generated a system that combines click chemistry together with the PISA, and we see that the system actually adapts. You can see that in these two papers with Pierce and myself in Central Science in 2021, and with Pierce, Jenny Yulin, and myself in Nano Letters a couple of months ago. What we do see is that life, far from being super complicated in its essence, it is possible to build systems which actually behave like life, but with a much more simpler chemistry. So my summary and conclusions are here. We will talk much more later if you want to, and I will also be engaged in the internet answering questions you may have. We have the following conclusions. Natural living systems share a set of 4 properties. With those 4 properties, one is able to actually write some form of equations. These properties are unified in the universal cell division cycle. And they are in natural life, they're expressed with biochemistry, which operates in water. But life, when it was very early in the planet, in the history of the planet, probably was far simpler. And there is a lot of evidence later in the history of life, there is a lot of evidence that that's the case. But what does it take to make a self-booted, fully chemically operated, synthetic, that is to say, without using any biochemistry living system? Well, that's a question we have asked and we have answered for a type of systems. So life, for that, we have used the fact that life's properties can be unified and represented by a simple set of stochastic differential equations. And their solutions, which definitely are solutions to equations that are not biochemistry or anything that are not representing biochemistry. Those solutions have, with the correct boundary conditions, have behaviors that are seen in very simple living things themselves. And the solutions can be represented through vesicles or coacervates, another possibility. Both offer a free energy gradient. Using PHOTO-PISA, photopolymerization-induced self-assembly, in a one-pot chemical implementation, which starts with something which is homogeneous, you can translate the solutions by first generating the vesicle, generating the membrane. And this requires a few simple and non-biochemical compounds which can generate these amphiphiles in water. The amphiphiles will self-organize and they autonomously boot up as self-organized dynamical because they change, they evolve with time, which evolve into a population of biochemistry-free vesicles that one handle information, the one that was in the molecules that were put in the initial solution plus the environment. They metabolize, that is to say, they make their own parts and they harness the energy from the environment. They self-reproduce, as you saw. They struggle for existence, they compete. And something I didn't discuss in detail, but they also do, is they actually provide inheritance, variable inheritance. Not all the molecules which are exchanged, all these partially reactive amphiphiles have the same lengths. Their polydispersity index is about 1.1, 1.2, and that leads to heritable variable inheritance, which is very important. The heritable variation is very important for Darwinian evolution. We have also shown that they adapt by combining PISA with click reaction. And we conclude that, as said here in 5, that life is a consequence of carbon chemistry and the physics of self-assembly, as was said by Stephen Jay Gould in some editorial in the New York Times many years ago, for some reason that if anybody asks, I will explain a little bit. And this is a form of what Bernard called generalized life, or life beyond biochemistry, as a colleague of myself and myself are now calling this. This has implications for artificial construction of synthetic living systems, for connecting life and polymer science to interstellar medium chemistry, the chemistry of the interstellar medium field, which is booming, and life in exoplanets also, and in the universe. The conditions in all the places are different. Why not starting with some protocells like we did here and arrive to life as we know it today, or life as it was here 3.6 billion years ago. So for the origins of life and the study of the protocell era on Earth, and in general to understand if life is a consequence of the evolution of the universe. Finally, it is very easy to implement. This system is very easy to implement, it's cheap to implement. You do need some sophisticated facilities for doing an MR and so on and so forth, but the experiments themselves are easy to do and is easy to implement by undergraduates or graduates or anyone who has curiosity. And the question next is not what is life, but why life? Why does life exist on this planet? That is not answered, but experiments and systems like this can help answering it. And I am finishing that part of the presentation by thanking the people who sponsor me, Repsol, an oil company from Spain, the university Harvard, the Harvard Origins of Life Initiative, a foundation is called Everest, and the Center for Nanoscale Systems of Harvard University, which is funded by the National Science Foundation. Thank you very much for your time and attention. I hope I didn't bore you. And if you have additional questions, want reprints, comments, whatever, please email me at this address, jperezmercader, all together,